Hi, can somebody confirm you can hear me? I'm getting an
Hear
error
you.
message saying my speaker isn't working.
I can hear you. We hear
Good.
you.
Sorry for being a bit late. When I tried to log in today through the security system, I had to get a call from Microsoft and the phone call never came. So I had to redo it, and unfortunately I got the text message, but not the phone call. Any questions before we begin? If not, let me talk a little bit about the grading. I meant to talk about that last time, um, last Tuesday. I did get the extra credit quiz, practice quiz 00 graded. So you can see that. Let me see if that grade shows. In the past, that grade doesn't show unless you click on it. This time it looks like it's showing. Quiz 1C and 2C are graded. I've not graded the extra credit picture. I have, however, graded extra credit study week 2 and extra credit study 3. I've graded up to lecture worksheet 3. And I've graded lab 4 for the first time, as well as lab 3 for the first time. Let me check on lab 3. Yeah, you may put in a correction up until August 3rd at 11:59 p.m. for lab 3. And that's because I didn't get it graded early last week. I got it graded fairly late last week. And so I decided to just move it to this Saturday. And then lab 4, I think is, I graded at the correct time. And it's going to be due for the second grading on August 3rd as well, this Saturday. I have not graded lab 5, and so its second grading will not be after August 3rd. It'll be after, I guess I can change that right now. It will be after, whatever that be, August 10th. Let me remind you, if you don't get 100% in the lab on the first grading, you can turn in the lab again for a second grading for full credit. Oh, here we go. I forgot they changed that. Looking for the date, and you don't see the date anymore. You have to click on Manage Due Dates and Assignment, and then it'll show you the dates. So just change that to August 10th for lab. Looks like I changed that for the wrong lab. Nope, that's supposed to be lab 5. So I did it right. Save that. All right, any questions about the grading or anything else? If not, let me get out of the grade book and then I'll share my screen.
I have a question regarding the unknown project. Should we
Uh-huh.
expect that number from you over the weekend, or hasn't it come through yet, correct?
I have chosen a number for you guys, but I haven't mailed it out yet. I'm just going to mail it all together on Tuesday with your first results for the gram stain, the gelatin hydrolysis, and the PR lactose test. So I'm just going to email it all together.
Perfect. I just want to make sure I didn't miss it. Thank you.
No, you haven't missed it. All right, let me share my screen now. Oh, that won't work. Got to go to Zoom to do that. It's not showing unless I blow it up. All right, you should be seeing my screen now. So today is August 1st, and we are supposed to be working on chapter 5 and actually starting chapter 6. Is that right? I don't think we'll get there, but we'll try. Any questions about what we're doing? All right. Um, Someplace I got that. Is this the slide that we stopped at last time?
Yes.
Great. Thank you. So we had talked about the nutritional classification system, and you can separate all organisms based upon where they get their energy source and where they get their carbon source. Um, so we can break them into the four groups: the photoautotrophs, the photoheterotrophs, the chemoautotrophs, and the chemoheterotrophs. When we're talking about energy production, you should realize that nutrient molecules have energy associated with the electrons that form their chemical bonds. In cells, energy is obtained from catabolic reactions and stored in the bonds of ATP. And then when the cell needs energy, it breaks apart ATP, which releases energy, and that energy can be used for the cell's needs. ATP is called the energy carrier of cells because when cells need energy, they tend to get it from ATP. ATP has a high energy unstable chemical bond, and you break off the third phosphate, it'll release a lot of energy. That's one of the high energy unstable bonds. And then energy is released quickly and easily, and the cell can use much of that energy for the cell's energy needs. Any question about any of that? When we're talking about energy production, you should realize that cells ultimately obtain their energy needs from carrying out oxidation reduction reactions. Oxidation is the loss of electrons. Reduction is the gain of electrons. So when a molecule A loses an electron, we say it becomes oxidized. And then when molecule B gains the electron, we say molecule B becomes reduced. Oxidation reduction reactions are always coupled. And we have a shorthand for saying oxidation reduction reactions, and that is a redox reaction. An exact example of that would be copper plus 1 gets together with iron plus 3, and then copper loses an electron, becomes copper 2 plus. Iron gains the electron, becomes iron 2 plus. Iron is reduced, copper is oxidized. Any question about that? Now here's an easy way to remember oxidation reduction reactions. And that is to say, Leo the lion goes grr. Leo, lose electrons, oxidation. Grr, gain electrons, reduction. Any question about that? That's the mnemonic device that I even use today. In biological systems with oxidation, the protons, meaning H plus ion, are usually removed with the electrons. Remember, a proton and an electron equal a simple hydrogen atom. And therefore, biological oxidations are often called dehydrogenations. And that's because when the electron is lost by a molecule, the hydrogen ion is typically lost by that molecule also. And then where the electron goes to reduce that molecule, the hydrogen ion tends to go as well. Any question about that? And an example of this, I thought I had that example there. An example of this is NADH. When it loses an electron, oh, excuse me, this is, I got it wrong here. There's NAD plus there, so it's gaining the electron. So this would be something like glucose giving off its electrons to NAD plus. The hydrogen ions tend to also leave the glucose and then move to the NAD plus. And then the NAD plus is oxidized, gaining an electron and a hydrogen ion, and becomes NADH. And actually NAD plus gains 2 electrons and only 1 hydrogen ion. Any question about any of that? All right. Biological redox reactions, remember that's oxidation reduction reactions, are used in catabolism to extract energy from nutrient molecules. We have highly reduced compounds like glucose, which will be degraded or catabolized to a highly oxidized compounds. A reduced compound will be a compound with many hydrogen ions, and glucose has many hydrogen ions. If you don't know, you should know or learn this. The glucose is C6H12O6, a highly reduced compound with many hydrogen ions, and glucose will be oxidized to CO2 and water, which obviously has less hydrogen ions, at least on the CO2 part, and it's highly oxidized. Much less energy in this than in the glucose. Some of that energy, which is released by glucose in oxidation reduction reactions, can be stored and trapped in the chemical bonds of ATP. So highly reduced compounds like glucose contain a large amount of potential energy, and that energy can be used by the cell to make ATP. So let's look at aerobic respiration, the main way our cells obtain or make ATP. We start with glucose and oxygen, and in a summary reaction in cellular respiration, also called aerobic respiration, those two terms are used synonymously. I tend to use aerobic respiration because there is respiration that can happen in a cell and it's not aerobic respiration. And you just have to remember when they use cellular respiration, they mean aerobic respiration. They do not mean anaerobic respiration, which is respiration happening in a cell. Okay, so the energy from glucose will be released. Some of that will be converted into heat. Some of that will be converted into the energy and stored as ATP. And then the glucose and the oxygen will be converted to carbon dioxide and water. So this is an overall summary reaction with the chemical reaction shown here. C6H12O6, that's glucose, plus 6 oxygen molecules, plus 38 ADP molecules, and 38 phosphate, that's inorganic phosphate, generate 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any question about any of this slide, any of this summary equation for aerobic respiration? We will talk about aerobic respiration in detail in just a few minutes. So when we are making energy, most of that energy that the cell uses for its own energy needs will be stored as ATP. So some of the energy from some redox reactions is used by the cell to make ATP. Remember, with animal cells, especially human cells, but any animal cell, there's really only two redox reactions which can make ATP, and that would be aerobic respiration and fermentation. Even in green plants and green photosynthetic bacteria, there are only a few catabolic reactions that can be used to make ATP. It's more, in fact, many more than animal cells, but it's still only a few. Most catabolic reactions cannot generate ATP. ATP is generated by phosphorylation of ADP. Phosphorylation is simply the addition of a phosphate group to a molecule. And so we put this phosphate group, it's actually an inorganic phosphate group, onto ADP to make ATP. And so we have ADP here. We get energy from an aerobic respiration or fermentation, and that will be in oxidation reduction reactions to generate ATP. You'll note, I've already mentioned it before, that ATP has some high energy bonds. The third phosphate group is a high energy bond, and that's shown by a squiggly line here. The second phosphate is also a high energy bond. And if the cell needs energy, it can break off that third phosphate group. And if the cell needs even more energy, it can also break off that second phosphate group. Any question about any of that? When we're generating ATP, cells have three mechanisms for making ATP. The first is substrate level phosphorylation, where you have a molecule with a phosphate already bound to it, and you then transfer this phosphate group from some molecule to ADP, and then that makes the molecule minus the phosphate, and then ADP is converted to ATP. This actually happens in fermentation and glycolysis, and we'll look at that in just a few minutes. The second way that cells can generate ATP is by respiration. And respiration has a couple of different names, like oxidative phosphorylation. I'll always use the term respiration. Note how I'm not saying aerobic respiration. When we use the term respiration, we mean all forms of respiration. So that would include aerobic respiration and anaerobic respiration. In respiration, we have the sequential transfer of electrons from an electron donor molecule, such as glucose, to a intermediate molecule, which transfers it to another intermediate molecule, to many different intermediate molecules, until the electron is transferred to the final electron acceptor. That is the last molecule to accept the electron. In aerobic respiration, electrons are donated by glucose, and the final electron acceptor of aerobic respiration is oxygen. And that's shown here, where the electron leaves this molecule and then combines with oxygen. Oxygen, the electrons, and the addition of hydrogen generate water. And we'll talk about that later when we're talking about aerobic respiration. Along the way, energy is released in controlled steps. And some of these steps, ATP can be made. Any question about any of that? All right. The third way that cells can generate ATP is by photophosphorylation. Photophosphorylation only happens in photosynthetic cells, such as green plant cells, algae cells, and photosynthetic bacteria. Light releases electrons from chlorophyll, and then the light energy is converted to chemical energy via an electron transport chain. And we'll talk about an electron transport chain in just maybe in a half an hour. Along the way, ATP and NADPH are generated by photosynthesis or photophosphorylation. However, the ATP and the NADPH are often used to make sugar. Any question about any of that? And this is showing you a picture of photosynthesis, which explains why the boys are so muddy. Any question about any of that? Actually, the boys don't engage in photosynthesis. Only the plants do in the background, and any photosynthetic microbes in the background. All right, returning to the production of energy, cells have metabolic pathways to make energy. The energy is released from molecules in controlled ways and stored by a series of reactions, meaning the energy is not released all at once. Like when we burn glucose, the energy is not released all at once, and it is done in a stepwise fashion where the energy is released in controlled ways, and that will keep the cell alive, because if you were to release the energy from glucose all at one time, that would fry the cell. That would be releasing too much energy and heat, and that would kill the cell. Electrons from donor molecules are passed from one compound to another through a series of redox reactions. And where do I have that? Right here. Where the energy initially from glucose is passed to NADH, which then passes it to this molecule, which passes it to that molecule, to that molecule, to that, to that, to that, to that, until it's finally passed and loses, the electrons lose that molecule, and then are passed to oxygen in aerobic respiration. And those are a series of redox reactions. Any question about any of that? So NADH, which loses the electron, will become oxidized. FMN, which gains the electron, will be then reduced. And then FMN, you don't need to know that, it will lose the electron and become oxidized. Any question about any of that? All right. So when we're talking about making ATP, ATP is frequently made from carbohydrate catabolism. In our cells, this is the only way that our cells can generate ATP from carbohydrate catabolism. Most organisms use as their primary source for cellular energy carbohydrates, particularly glucose. The most common energy source is actually glucose. But cells can use other molecules like lipids and proteins and degrade them to make ATP. We will talk about that near the end of this lesson. To produce energy from glucose, microorganisms use two general processes: respiration and fermentation. When I say respiration, I mean both aerobic respiration and anaerobic respiration. Our cells can only generate ATP from glucose using aerobic respiration or fermentation. But microbes can use anaerobic respiration. Let's watch a little video of aerobic respiration. Assuming that it's going to load. Oh, there it goes, just very slow. This is extremely slow. I think I'm going to continue discussing and we'll come back to this slide. Aerobic respiration is one way that our cells can generate ATP. The only other way our cells can generate ATP is by fermentation. You blow that up so you can see this a little better. Now remember, particularly bacteria cells can use either aerobic respiration or fermentation, but they may also use anaerobic respiration. We're looking at aerobic respiration on the left and fermentation on the right. Both of these processes prefer using glucose as a starting molecule to generate ATP. And aerobic respiration has three principal stages: glycolysis, the Krebs cycle, and the electron transport chain. Essentially, electrons flow from glucose, which is an energy-rich molecule, and glucose will be converted to CO2 and water, which are energy-poor molecules. And along the way, ATP will be made. In aerobic respiration, we get a high yield of ATP. On the right, we have fermentation, which always starts with glycolysis. And then you attach one or two fermentation steps after the glycolysis. And so fermentation has glycolysis, but no Krebs or electron transport chain. And glucose will be converted to the end product of fermentation. It will not be converted to CO2 and water. And the end product will depend on what fermentation reaction we're talking about. We'll only discuss, well, mainly two, and that is lactic acid fermentation, where the end product will be lactic acid or lactate, and then alcoholic fermentation, where the end product will be ethanol. There's a much lower ATP yield from fermentation than aerobic respiration. Any question about any of that? Finally, this loaded. I don't know why it's so slow today.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
From idea to submission, empower students to do.
Okay, any questions about that video? Come on, there we go. So this slide is showing you an overview of aerobic respiration. This would be aerobic respiration in a eukaryote cell. The cell is shown here by the purple square. And aerobic respiration has 3 parts: glycolysis, the Krebs cycle, and then the electron transport chain. And these parts are shown here. Glycolysis. The citric acid cycle is also called the Krebs cycle. We'll talk a little bit more about that. And then the electron transport chain is right there. The point is that glycolysis happens in the cytoplasm of the cell. During glycolysis, glucose is converted into pyruvate, also called pyruvic acid. Some ATP is generated, shown here. Come on. And NADH is generated. The purpose of NADH is to carry electrons from glucose to the electron transport chain, and we'll come back to talking about that. The pyruvate then in a eukaryote cell enters the mitochondria and then will perform the preparatory step in the Krebs cycle inside the matrix of the mitochondria. That will mean inside the inner mitochondrial membrane. That's what we mean by the matrix. In the Krebs cycle, we do generate some ATP. We also, in the preparatory step and the Krebs cycle, so I guess the preparatory step is actually shown right here. And I'm not sure why they're trying to make it there, because that happens here. But CO2 is generated as well as ATP. And then a whole bunch of NADH2 and some FADH2 is made by the preparatory step and the Krebs cycle. The purpose of NADH and FADH2 is to take the electrons, which ultimately came from glucose, and then take those electrons to the electron transport chain. The electron transport chain will take these electrons and then use them to make ATP in oxidative phosphorylation, and it'll be a high yield of ATP being produced at this stage of the electron transport chain. The electron transport chain happens across and in the inner mitochondrial membrane. So the membrane of the mitochondria. Any question about any of that? All right. The first step of aerobic respiration, and actually the first step of anaerobic respiration as well, is glycolysis. Glycolysis does have another name. I don't use it. Your textbook tends not to use it either. Embden-Meyerhof pathway. In glycolysis, we have the oxidation of glucose, and that's converted to pyruvic acid. Pyruvic acid and pyruvate are essentially the same molecule, and your cell will use one like the other. You need a biochemist to detect the difference. The only difference is pyruvate has the hydrogen added on it, and that we call pyruvic acid. Any question about any of that? So in glycolysis, we have the oxidation of glucose to pyruvic acid. It occurs in most living cells. It can occur in the presence or in the absence of oxygen. Glycolysis does produce some ATP and some NADH, shown here and there, or there. I guess we're talking about aerobic respiration, so it should be here. Although ATP is only over here, so you need it over here too. Any question about glycolysis? Here's the chemical reactions of glycolysis, starting with glucose here, ending up with pyruvic acid here. And you'll note that pyruvic acid is a 3-carbon molecule. Glucose is a 6-carbon molecule. And so in glycolysis, we'll generate 2 pyruvic acid molecules per molecule of glucose. And that's actually shown here. What you do is you make this. Come on here. You make this, and then you make that can be converted into that, and then can go down twice. So we could have just drawn this whole thing, connecting it right there. The important part is you get 2 pyruvic acids from 1 glucose. You get 4 ATP made by glycolysis, 2 here and 2 there. However, you have to put in 2 ATP in glycolysis. So out of glycolysis, you only get a net gain of 2 ATP. Any question about any of that? All right. When I was an undergraduate, I had to learn all of the intermediate steps of glycolysis, as well as all of the enzymes for performing each step. And since I had to learn all the names and parts, I think it's fair that you guys have to learn that as well. What do you guys say to that?
Maybe not.
Maybe not. You do need to know the summary of glycolysis, shown in red here, but you start with glucose, and then you end up with 2 pyruvates, also called pyruvic acid. You get a net gain of 2 ATP and 2 NADH out of glycolysis. And so that would be the equation up here, making this right there. You don't need to memorize this equation. Any question about any of that? All right. So we're talking about aerobic respiration, but I want to talk about respiration in general. And that is, respiration is the oxidation of molecules, which releases electrons, and then these electrons are used by the electron transport chain to generate ATP. There are two forms of respiration: aerobic respiration, where the final electron acceptor is oxygen. So oxygen is where the electrons end up in aerobic respiration. In anaerobic respiration, the final electron acceptor is some other molecule other than oxygen. And that is just the definition of anaerobic respiration. It depends on which form of anaerobic respiration it is, what that molecule will be. For example, that molecule could be sulfate, SO4 with a negative charge, or it could be hydrogen sulfide gas, H2S. And those are just two different types of anaerobic respiration. And it could be nitrate or some other form of anaerobic respiration. In respiration, ATP is generated by oxidative phosphorylation. And that's respiration in a nutshell. Returning to aerobic respiration, after glycolysis, we have the Krebs cycle. But we have one step before the Krebs cycle called the preparatory step. Pyruvic acid cannot directly enter the Krebs cycle. It needs to be converted from pyruvic acid into acetyl coenzyme A. And so the preparatory step changes pyruvic acid into acetyl or acetyl coenzyme A. Both pronunciations are correct, by the way. During the preparatory step, we generate one NADH molecule, and we also generate our first CO2 molecule, meaning pyruvic acid is converted in the preparatory step to acetyl coenzyme A. Pyruvic acid is a 3-carbon molecule. Acetyl coenzyme A is a 2-carbon molecule. 1 carbon comes off and will be the first carbon coming off as CO2 for all of the carbons in pyruvic acid, or actually all of the carbons in glucose. All right, for each glucose molecule, the preparatory step will make 2 molecules of acetyl coenzyme A. Can anyone explain to me why in the preparatory step we generate 2 acetyl coenzyme A's and not 1 or not 3? All right, I was hoping somebody would get that. The reason is, per molecule of glucose, we will generate 2 pyruvic acids in glycolysis. That means the preparatory step will run 2 times. For the first pyruvic acid, and then it'll run again for the second pyruvic acid. Any question about any of that? All right. So the preparatory step converts pyruvic acid into acetyl coenzyme A. Acetyl coenzyme A can then enter the Krebs cycle. The Krebs cycle has some other names. I don't use them, and your textbook doesn't use them often, but it does at least discuss them. And that is tricarboxylic acid, or TCA cycle, and also the citric acid cycle, which the citric acid cycle is actually abbreviated TCA as well. The T for the, C for citric, and then A for acid. Any question about any of that? I will always call it the Krebs cycle. Your textbook tends to call it the Krebs cycle as well. It's called the Krebs cycle just because Dr. Krebs is the person who discovered it. During the preparatory step and the Krebs cycle, we generate one ATP molecule, shown down here, 4 NADH molecules. The first in the preparatory step right there, the second right here, the third right there, and then the fourth right there. We generate one FADH2 molecule right here. Remember, the FADH2 and the NADH2 are destined to carry the electrons that ultimately came from glucose to the electron transport chain. And then the cycle ends with the formation of the molecule that began the cycle, meaning acetyl coenzyme A enters the Krebs cycle and it combines with oxaloacetic acid to make the first molecule, which is citric acid. You don't need to know that. Okay. Need to turn my phone off. It keeps ringing. And then the molecule is changed to here, which 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 is changed to oxaloacetic acid. And this is the molecule we started with. And that's why it's called the cycle. Because when the Krebs cycle is running, it ends up with the molecule we start with, meaning the Krebs cycle will continue in your cells for as long as you are alive. Any question about that? And I suppose you have to say, as long as you're alive and you have a source of glucose, if you're starving, you may not have the Krebs cycle going on, but generally the Krebs cycle will be running all the time in your cells. Now remember, glucose in glycolysis makes 2 pyruvic acids. So the preparatory step and the Krebs cycle will run 2 times for each molecule of glucose. So if I ask you how many ATP are made by the Krebs cycle, the correct answer would be 1 right here. But if I ask you how many ATP are made per molecule of glucose in the Krebs cycle, you would have to multiply that by 2, and then the correct answer is 2 ATP. Any question about any of that? All right. Oh, wait, I gotta mention something else. I had it up there too. I'm just not sure why I didn't mention it. In the preparatory step and the Krebs cycle, each of the carbons that came from pyruvic acid will be burnt to CO2. So pyruvic acid has 3 carbons. The first one will be made in the preparatory step, come off as CO2, and then we have another CO2 generated here, and a third CO2 generated here. So each carbon in pyruvic acid is burnt to CO2. And of course, we have 2 pyruvic acids coming from 1 glucose, and that means each carbon in glucose will be converted or burned to CO2. Any question about any of that? The importance of that is when we breathe out CO2, this is where that CO2 came from. The preparatory step and then the Krebs cycle, converting the carbon in glucose into CO2. None of the oxygen you breathe in is converted into CO2. All the CO2 we breathe out comes from aerobic respiration. Any question about any of that? All right, looking at the overview of aerobic respiration and fermentation, we're over our next step of aerobic respiration, which is the electron transport chain. The electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as electrons, which flow from glucose, are passed down the chain. Energy is released as the electrons are transferred from a high energy to a lower energy compound. And that energy is released by chemiosmosis. Let me just end this and show you the electron transport chain one more time. No, there it is, right there. It's the electron transport chain. And we'll look at that in a little more detail in just a few minutes. Let's just watch a little video on the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation.
Okay,
This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Any questions about that video? All right, let's look at aerobic respiration in a little more detail. Aerobic respiration has the third step of it as the electron transport chain. The electron transport chain occurs along and in the inner mitochondrial membrane, obviously of the mitochondria. What it is a bunch of proteins embedded in a membrane, and the electrons from NADH and FADH2 are brought to these proteins, and then the electrons move from one protein to another until they leave the proteins and will join with oxygen. We'll talk about that in a little bit. The important part is this is happening in a bunch of proteins in a membrane, and that in eukaryotes is the inner mitochondrial membrane. In prokaryotes that don't have a mitochondria, this occurs in the cell membrane. Any question about any of that? So we have similar, if not the same, proteins in prokaryotes. It's just that they're in the cell membrane, not the inner mitochondrial membrane. The point of having the electrons move across these proteins is that when the electrons move across some of the proteins, this protein here, this protein here, and that protein here, they give this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. These hydrogen ions create a potential energy difference from this side of the membrane to that side of the membrane, meaning there will be more hydrogens on this side of the membrane than that side. There will also be more positive charges on this side of the membrane than this side of the membrane. These positive hydrogens will want to, by the laws of diffusion, move to this side of the membrane. However, they cannot move across the lipid bilayer of the membrane, except at this channel protein, which is an ATP synthase enzyme. And as the hydrogen moves through this protein, it gives this protein, come on mouse, it gives this protein the energy the protein needs to phosphorylate ADP and create ATP. That energy is coming from the potential energy difference of the hydrogen ions on this side of the membrane versus that side of the membrane. Any question about any of that? And there's one other equation happening in this picture, and that is the electrons, which ultimately came from glucose and are being carried and brought to the electron transport chain by NADH, shown here, and FADH2, shown here. These electrons will flow through these proteins in the membrane and then leave the last protein and join with oxygen, which oxygen will join with the electrons and join with hydrogen ions to form water. And that's why you have to breathe in oxygen. All of the oxygen you breathe in will be converted into water in aerobic respiration. None of that oxygen is converted into CO2. Any question about any of that? In the electron transport chain, ATP production is kicked into high gear. In eukaryotes, we produce 32 to 34 ATP molecules in the electron transport chain of aerobic respiration. In prokaryotes, they're a little more efficient. They produce 34 molecules of ATP per molecule of glucose. Any question about that? No question. Well, why is it in eukaryotes sometimes 32 and other times 34? It's because 3 cells in your body can generate 34 molecules of ATP in the electron transport chain, and all the other cells in your body can only produce 32 molecules of ATP in the electron transport chain. The 3 cells which are more efficient are the heart, the liver, and the kidney. And why do you think the heart, the liver, and the kidney are more efficient than your brain cell, or your skin cell, or your nose cell? What do you know about the heart? Come on, somebody has to know something about the heart. I could ask, what do you know about the kidney? But I'm sure most of you wouldn't know about the kidneys. What do you know about your heart? What's the heart always doing?
Pumping blood.
Yeah, it's always working. It is always working as long as you are alive. It's not until you're dead that the heart ever stops working. That isn't true for all the other cells in your body, like your skin cell or your brain cell, which should be obvious since a lot of you didn't even guess. And that is, your brain cell is not always working. So the heart, the liver, and the kidney, these cells are always working. The kidney is always purifying the blood, removing nitrogenous waste from the blood, storing it as urine, and then putting it to the bladder. And the liver is always filtering your blood, looking for toxic substances, and then usually metabolizing it. So the liver too is always working. The liver does a bunch of other work, like it stores down glucose as glycolysis. Is that right? No, glycogen. Sorry, glycogen. Stores down glucose as glycogen. And then when your blood needs more glucose, the liver will release the glucose from glycogen into the blood. So the liver does a bunch of work. My point is, the liver, the heart, and the kidney, they're always working. And so these cells are more efficient than all your other cells. And the liver, the heart, and the kidney can produce 34 molecules of ATP per molecule of glucose. All the other cells in your body can only produce 32 molecules of ATP. And prokaryotes are just a little more efficient. I've already mentioned in aerobic respiration, the final electron acceptor is oxygen. Oxygen combines with hydrogen and the electrons to form water. And that's what happens to all the oxygen you breathe in, which is shown here. We'll go through this in a little more detail. This is the flow of the electrons in the electron transport chain, where the electrons will flow to the final electron acceptor, which is oxygen. You don't need to know any of these molecules. We'll talk a little bit about cytochrome C, but all the others we don't talk about. And this is the redox reactions happening here, controlling the release of energy from glucose. Because if glucose were to release all the energy at one step, that would fry your cell, kill the cell. Too much heat being released from glucose. and showing you the electron transport chain right here, which is a lipid bilayer. For a eukaryote, it is the inner mitochondrial matrix. It's the cell membrane for the prokaryote. And the point of this is when the electron flows through some of these proteins, this one here, that one there, this one here, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. Now, in a eukaryote, those hydrogen ions end up between the inner membrane and the outer membrane. And so in eukaryotes, we have our mitochondria, which protects the rest of the cell. In prokaryotes, these hydrogen ions are actually pumped into the cell wall, assuming the bacteria has a cell wall. And those hydrogen ions come through this ATP synthase channel protein, which will use the flow of the hydrogen ions to phosphorylate ADP to make ATP. I've kind of already stated that the flow of electrons to this side of the membrane causes a gradient. There's more hydrogen and more plus charges on this side than that side. That creates potential energy difference. That potential energy is used to make ATP. You don't need to know it. It's in chemoosmosis. It's a term I don't use, and that's the flow of the hydrogen to make ATP. The electron transport chain is sometimes called oxidative phosphorylation. I don't use that term because it's so different than the electron transport chain. So the complete oxidation of glucose via aerobic respiration yields 36 to 38 ATP molecules in aerobic respiration, 2 in glycolysis, 2 in the preparatory step in the Krebs cycle, and the remainder will be 32 to 34 in the electron transport chain. In prokaryotes, they're a little more efficient. They always generate 38 ATP. So here's the overall summary equation for aerobic respiration. We have glucose, C6H12O6, which combines with 6 molecules of oxygen and 38 ADP molecules and 38 inorganic phosphate molecules to generate 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. You do need to know this overall summary reaction as well as the summaries for each of the steps of aerobic respiration. All right, so this is looking at the summary of aerobic respiration here. On the left, showing glycolysis, starting it off, the preparatory step in the Krebs cycle shown here, generating NADH and FADH2, which carry the electrons to the electron transport chain, which generates 32 to 34 ATP, for a total production of aerobic respiration for 38 ATP in aerobic respiration, because we have 34 from right here, at least talking about eukaryotes, heart, liver, and kidney cells, 2 from the Krebs cycle, and then 2 ATP from glycolysis. Any question about any of that? And then one other thing, or actually two other things, is that this shows you the generation of the CO2 from each carbon of glucose, meaning we generate 6 CO2. And then all of the oxygen you breathe in will be converted into water. Humans aren't terribly good at storing that water, but desert animals are. And desert animals get about 65% of their water needs from aerobic respiration, where they breathe in oxygen and then generate water. Any question about any of that? All right. See how we're doing time-wise? Oh, we got lots of time. So that was aerobic respiration. Now let's talk a little bit about anaerobic respiration. Anaerobic respiration has a final electron acceptor in the electron transport chain, which is a molecule other than oxygen. It's some inorganic molecule, such as nitrate or nitrite or sulfate or iron. And the electrons just flow to this inorganic molecule. Anaerobic respiration tend to have both glycolysis and the Krebs cycle. However, their protein chain of the electron transport chain is shorter in anaerobic respiration than aerobic respiration. And because the electron transport chain of anaerobic respiration is shorter, it will yield less ATP than aerobic respiration. The yield of ATP will depend on which form of anaerobic respiration we're referring to, like nitrate anaerobic respiration differs on the yield of ATP than sulfate anaerobic respiration. And the yield will always be something a little bit greater than 2 to 36 molecules of ATP per molecule of glucose. And so anaerobic respiration is not as efficient as aerobic respiration. Even the very best, I used to know what that was. It's not nitrate or nitrite. I don't remember what that is anymore. But the best form of anaerobic respiration can only generate 36 molecules of ATP, which is less than aerobic respiration. And therefore, anaerobes tend to grow more slowly than aerobes. And it's simply because they generate less ATP per molecule of glucose. Any question about any of that? All right, so that's it for anaerobic respiration. Really, all you need to know is that the electrons end up at a different molecule than oxygen. Oh, let me show you a chain. Here we go. This is the electron transport chain of, actually this right here is the electron transport chain of aerobic respiration. And you can see the last protein is one that pumps hydrogen from this side of the membrane to that side of the membrane. And if the anaerobic respiration is a smaller electron transport chain, they will be lacking at least that protein. The worst forms of anaerobic respiration will only have this one protein. They won't have any others. and they will generate something like 4 ATP molecules of ATP per molecule glucose. You don't need to know that. You just need to know it's something between 2, a little greater than 2, and 36 molecules of ATP per molecule of glucose. All right, so that's anaerobic respiration. Let's talk a little bit about fermentation. That is another way that cells can generate ATP from carbohydrate metabolism. Our cells can generate ATP from aerobic respiration, and some of our cells can generate ATP from fermentation. Does anyone know what cells in your body can generate ATP from fermentation? Nobody knows that? You should know that, by the way.
Is it muscle cells?
Yeah, your muscle cells can generate ATP by fermentation. They only do that when oxygen becomes limiting, meaning your muscle cells, like if you're a couch potato, like most of the time me, your muscle cells will perform aerobic respiration to generate ATP. And that's because it's more efficient, a lot more efficient than fermentation. But if ever the muscle cells become limiting on the availability of oxygen, the muscle cells will fall back on fermentation. The reason why your muscle cells do that is the heart and the lungs are working like mad to supply the muscle with blood, which will deliver oxygen to the muscle, but the muscle is working so hard that it's used up all of the oxygen. And therefore the muscle will fall back on fermentation so that the muscle cell can continue to work. If the muscle cell has no glucose or no, is it NADH? No NAD plus, the muscle cell cannot perform fermentation and it cannot make ATP. And then what will happen to that athlete when their muscle cells can no longer make ATP? I don't know if it's happened in this Olympics or not, but almost in every Olympic, you have one Olympic athlete that pushes themselves really beyond their limit, and their muscles use up all the oxygen. So then they rely on fermentation, and then the muscle cell uses up all of the glucose in the muscle. The muscle cell can no longer make ATP, and then the Olympic athlete collapses because their muscles can no longer work. In the wintertime, usually their coach runs out and covers them with a blanket. In the summertime, their coach runs out and squirts them down with a bucket, not a bucket, but like a squirt gun or something with water, just to cool them. The Olympic athlete will then, their heart and their lungs will be working like mad, trying to supply the muscle with blood, which will supply the muscle cell with both glucose and oxygen. And then the Olympic athlete can stand because they can perform aerobic respiration of their muscle cells, so they can make ATP. The Olympic athlete will then stand and usually walk across the finish line. Anyone ever see that? Like I said, it usually happens once in each Olympic. Nobody remembers seeing that? I don't know if it's happened in the last Olympics, but usually in each Olympics, there's one athlete where that happens. Oh, let's go on. fermentation just does generate ATP, but not much. All of the ATP generated by fermentation happens in glycolysis. As you remember from aerobic respiration, glycolysis generates a net gain of ATP of only 2 ATP per molecule of glucose. Remember, fermentation has glycolysis as the first part of the fermentation, and then we have a fermentation step or two to convert the pyruvic acid to the end product of fermentation. And that end product could be lactic acid if it's lactic acid fermentation, or it could be ethanol if it's alcoholic fermentation, or it could be some other molecule for a different type of fermentation. We're not going to talk about the other types of fermentation. Fermentation always begins with glycolysis, where we start with glucose. We do generate NADH, and we do generate a net gain of 2 ATP and pyruvic acid. The NADH does not go to the electron transport chain. We do not have a Krebs cycle. The pyruvic acid will be changed by fermentation to the end product of fermentation. And then the NADH generated here will be used up to make NAD+. Why does fermentation not store the NADH in the cell and then just wait until the muscle cell gets oxygen? Because the muscle cell will get a lot more ATP out of aerobic respiration than it will get ATP from fermentation. So why does the muscle cell not just store that NADH? Anyone have a guess? It's actually shown right here. You need NAD+ in order for glycolysis to occur. And so fermentation recycles that NADH to regenerate NAD+ to allow glycolysis to continue. Because if NAD+ becomes limiting, the cell cannot engage in fermentation. And I've already told you, that's a muscle cell, the muscle cell cannot engage in fermentation, the muscle will stop working, and then the Olympic athlete will collapse. Fermentation does not require oxygen. It can happen in the presence or in the absence of oxygen. In our muscle cells, fermentation will only happen in the absence of oxygen. But other organisms can engage in fermentation regardless of whether oxygen is present or not. Like yeast, when you're making leavened bread, there is oxygen in the dough when you put the yeast in there, but the yeast can engage in fermentation and then produce CO2, which will cause the bread to rise. It's just an example that fermentation can occur in the absence or the presence of oxygen. It doesn't use the Krebs cycle or the electron transport chain. And then the final electron acceptor is pyruvic acid, which will be converted to the end product of fermentation. Let's talk about 2 of the common forms of fermentation: lactic acid fermentation, where in one step, pyruvic acid is converted into lactic acid. The lactic acid actually makes your muscles feel like they're burning. It's from the lactic acid changing the pH of the muscle. making your muscle feel like it's burning. Lactic acid is very common in many of the foods we use, which are fermented, like sauerkraut, yogurt, even chocolate. Most cheeses, like cheddar cheese, the cheese is made by lactic acid fermentation. Most of the food we eat is made by lactic acid fermentation. The lactic acid helps preserve the food. But lactic acid lowers the pH, which helps prevent the spoilage of the food. That's why yogurt does not spoil very easily, and you can keep it in the refrigerator for a long period of time. You don't even need to keep it refrigerated. You could keep it unrefrigerated. I'm not sure what the time span would be, but something like a day or two, something like that, and it will not spoil. Alcoholic fermentation is another common form of fermentation where pyruvic acid is converted into ethanol. This is a two-step process. And pyruvic acid is a 3-carbon molecule. Ethanol is a 2-carbon molecule. And so 1 carbon comes out as CO2 for each carbon in pyruvic acid, meaning there's 3 carbon, 1 comes off. In glucose, we get 2 carbons coming off per molecule of glucose, which is shown here. The carbon coming off on alcoholic fermentation is why we put yeast in bread to get leavened bread. The yeast will make the CO2, which will cause the bread to rise. We also use alcoholic fermentation to make beer, and the CO2 that's given off gives beer its fizz. When we make wine, we make the wine in an open barrel or an open casket, and the CO2 will vent to the air. So you don't get the CO2 in the wine. However, if you were to ferment we'll just say wine in the bottle, and it's a cap bottle, that CO2 would be captured and will create bubbles in that quote unquote wine. We don't call that wine, we call it champagne. And that's the only difference between champagne and wine. And that is the champagne is made in a bottle that has been capped. Any question about any of that? So alcoholic fermentation is most commonly seen when making beer or wine, or in making leavened bread, yeast raising bread. There are different fermentation in products. We're not really going to talk about them, and they can happen in more than one fermentation step. We've talked about lactic acid fermentation and alcoholic fermentation, which happens by the yeast. I will mention there is one cheese which is made not by lactic acid fermentation, and it's made by propionic acid fermentation, and that's Swiss cheese. Swiss cheese, or that fermentation, sheds CO2, and that's why the Swiss cheese has a hole in it, or holes in it. Okay. Some of these fermentation reactions are made to make chemical products for our use, like formic acid, succinic acid, butanol, acetone. Come on, mouse. I'm circling it, but I can't get my mouse to work. Not sure what happened to my mouse. There it is. Right there. Acetone. All right. You don't need to know any of these others. I'm trying to remember. I think it's this one, which can convert alcohol or wine into vinegar. All right, any questions about fermentation? This slide is just showing you the different fermentation end products, the use of the fermentation. You can see lactic acid fermentation's a lot of them. You don't need to know the starting material. And then the microorganism, you don't need to know that either. The microorganism performing the fermentation. Oh, there you go, vinegar. There's one here. Acetic acid, acetobacter, using ethanol or wine. All right. So here's a good summary of aerobic respiration, anaerobic and respiration and fermentation, comparing them together. When we look at the growth conditions, aerobic respiration requires oxygen, has to be present, or you can't get aerobic respiration to work. Anaerobic respiration does not require oxygen. It requires another molecule. And if the organism is a obligate anaerobe, it will require the absence of oxygen to perform the anaerobic respiration. For fermentation, this can happen either in the presence of oxygen or in the absence of oxygen. And so this fermentation can happen under aerobic respiration, under aerobic conditions or anaerobic conditions. The final electron acceptor, you don't really need to know this one, will be an organic molecule where pyruvate will be converted into the fermentation end product. You don't need to know this. And then the ATP molecules that generated, it's 36 to 38 in eukaryotes, 38 for prokaryotes. And then for anaerobic respiration, more than 2, but fewer than 38. So someplace between 4 and 36. And then the molecules of ATP made by fermentation is only 2. Any question about any of that? All right, apart from glucose, a carbohydrate, being used as a food source, microbes can also use other food sources. With carbohydrates, you simply break the carbohydrate down, and if needed, meaning break it down into simple sugars, and then if needed, convert that simple sugar into glucose. So for example, with lactose, you break lactose into glucose and beta-galactose, and the glucose you can use immediately, and then the beta-galactose has to be converted into glucose. And then you send the glucose down aerobic respiration in the normal way. Microbes can also use other carbohydrates such as starch. And then microbes can use other food molecules as well, including proteins and lipids. One problem with large molecules like large proteins and large lipids is this molecule cannot cross the cell membrane. And how both our cells as well as microbes handle the digestion of large molecules like proteins or lipids is they will break down the lipid or the protein outside the cell by secreting extracellular enzymes, which will then break down the lipid and the protein into smaller molecules or their monomers, which can then be transported across the cell membrane. For example, lipids will be broken. Well, if it's a fat, I got to say that, because if it's a steroid, I'm not sure how the cell digests that. But with a fat, it will be broken down into glycerol and the fatty acids. The glycerol will enter the Krebs cycle, and the fatty acids will be cleaved into 2 carbon chunks, which also enter the Krebs cycle. With the proteins, they'll be broken down into amino acids, which can be brought into the cell. The amino group will need to be removed from the amino acid, and then the remaining part of the molecule will be converted to enter aerobic respiration. Most will enter the Krebs cycle, but a few can enter glycolysis, and a few can enter the electron transport chain. I don't remember. I'll have to look that up. All right. So proteins will be digested. Even our cells do that. We secrete extracellular proteases, which will digest the proteins into amino acids. And then the amino acids can be brought into the cell and then used as a food source. And there you can see the proteins, depending on which amino acid they are. Some of them can be converted into glycolysis right here. Others will be converted into acetyl coenzyme A and enter the Krebs cycle at the very start of it. And then other amino acids will be converted into some intermediate in the Krebs cycle and enter late in the Krebs cycle. For lipids, they can be broken down into glycerol and fatty acids. The fatty acids will be separated into 2 carbon chunks, converted into acetyl coenzyme A, and then enter the Krebs cycle. The glycerol will be converted into a molecule of glycolysis. Any question about any of that? All right. So the body can digest lipids and proteins as well as complex carbohydrates. The carbohydrates, like I said, are fairly simple. You just break them down into simple sugars. If the simple sugar is not glucose, you just convert the sugar to glucose. And then send glucose down aerobic respiration in the normal way. Any question about that? Oh, I meant to say the body can digest amino acids or proteins and lipids and burn them in aerobic respiration. However, a marathon runner will not directly burn lipids until 45 minutes in the run. The first 45 minutes of the marathon runner's run, the marathon runner will not digest lipids and then burn them to generate ATP. It's only after 45 minutes that the marathon runner can do that. So I guess the important part is that the body can digest proteins and lipids. It's not the preferred thing the body wants to do. It will want to utilize carbohydrates first. Any question about any of that? All right, so we have the complete oxidation of glucose and aerobic respiration. And aerobic respiration is the most efficient way to make ATP. The energy in ATP is used for various cellular processes, such as active transport of a protein, for that active transport protein to move one molecule across its gradient, meaning against its gradient, and pump that molecule into the cell, will cost the cell 1 ATP molecule per molecule moved into the cell. The cell uses ATP for its movement, as well as the synthesis of new cellular components, meaning if we're making a larger molecule, the cell will need energy to make that larger molecule. The cell usually gets that energy from ATP. In fact, most of the cellular ATP is used for the synthesis of new cellular molecules, meaning the biosynthesis of carbohydrate, lipids, or proteins. Any question about any of that? All right. Let's talk a little bit about the integration of metabolism. So far, I've talked about anabolic and catabolic pathways as separate and independent events or pathways. They are not separate and independent. In fact, there are many common intermediates, and we can also reverse a catabolic pathway. And so instead of degrading a larger molecule and ending up with a smaller molecule, we'll start with a smaller molecule, which can be the same small molecule, and then build it up in an anabolic pathway. What I'm trying to tell you is that they are anabolic and catabolic pathways are joined by common intermediates in our cells. And humans are lousy at converting the pathways. Microbes are much better at it, so are green plants. But we are very good at doing it for the common biological molecules. We call these pathways that can work either way as a catabolic pathway or an anabolic pathway, an amphibolic pathway, a pathway which can run in either direction, meaning this way or that way. So a metabolic pathway that can function in both ways, as both catabolic and anabolic reaction, is called an amphibolic pathway. They bridge the pathways involved in lipid, carbohydrate, protein, and nucleic acid metabolism. So this slide is trying to show you the integration of metabolism. And as you can see, here we have glucose going down in glycolysis, going down in the preparatory step and the Krebs cycle, and then converted into CO2, which I guess would be shown here. Okay? However, we could start at the very end molecule, oxaloaxalic acid, and reverse the pathway. A double arrow is a reversible pathway. And we can go back all the way in the Krebs cycle back to acetyl coenzyme A. And we can convert acetyl coenzyme A into pyruvic acid. Furthermore, pyruvic acid can be converted in glycolysis, going reverse, into glyceraldehyde 3-phosphate. And you see how this is an arrow this way? That means we can start with this one and go that way. So this doesn't have the double arrow, but it is a reversible pathway. Why there's an arrow here and an arrow that way. So we can go with this to go here, and then go with this to go there, and then go with that to go there. Meaning we can convert any lipid into a fatty acid, make it acetyl coenzyme A, bring it up here, get the glycerol, bring it this way, go that way, and then go this way to make glucose. The same with protein. We don't have the protein here, but we can break it down into amino acids. And depending on which amino acid it is, we can go backwards to make glucose. And the same thing, you eat a whole bunch of carbohydrates. How do you store it as? We will convert that to acetyl coenzyme A, use acetyl coenzyme A to make fatty acids. We will convert other lipids. Where is that? Right here. Well, I guess we'll start here. Acetyl coenzyme A, convert it to here, to there, and then convert it to glycerol. And then this is a reverse pathway where glycerol can combine with fatty acids to make a lipid. And you all know that if you eat too much carbohydrates, what does our body do? We store that as fat, which is a lipid. The point is, even humans are very good at amphibolic pathways for the common biological molecules like glucose and lipids and proteins. In fact, if you were to eat a high fat diet or a high protein diet or both, your body will actually convert those molecules into amino acids for a protein and then go backwards in this amphibolic pathway to make glucose. If you're a couch potato, and then the body will use glucose, meaning glucose will be sent down aerobic respiration in the normal way to make ATP. Any questions about any of that? Oh, well, I think I'm going to stop right here. And we'll talk about photosynthesis in our next lesson. Any questions? If not, I'll see you at 7 for the lab. All right. Bye.